Oh my lord, that was weird. Okay. Let's see how that goes. Okay, so we're just gonna try to start. We're gonna try to finish this stuff up quickly, so there's time for me to kind of give you a peek into what we did in recitation today, which I'm hoping we'll get to after we make up recitation. Because today's recitation didn't have anything to make up, so we used it as a review. And I'm gonna post that stuff on Blackboard today so that you have access to it. But I would like to use the end of lecture today to kind of go over that stuff so we're all on the same page. So bear with me. Those of you who came to recitation today as an extra something to do, who still need to make up last Thursday's recitation, still need to come this Thursday to make that up, just so you know. Okay, other business, I finally successfully gotten out of Route 221. 275, we were in there today, it's awesome. It's only for Tuesday's recitation. Thursday, we'll be in 217 for both of my recitations. Dr. Texel's recitation does not change, except for this week. If you're in Dr. Texel's recitation, please come to one of my Thursday recitations. I can't say that enough times. I'm hoping everyone hears that. I keep getting emails that make me feel like I'm speaking unclearly. So come to my recitation. Don't go to Dr. Texel's recitation. She will not be there. She's not expecting you because I told her that you're coming to me because I want to do all the reviewy kind of things for you guys. Because what's the point of her doing a review if she's not teaching the class? Okay. Good? We happy? Yes.
So the recitation on Thursday, are we doing the paper that we missed from last week?
The recitation on Thursday is a makeup of last week. You're doing the paper from last week, and then we'll use the extra time at the end to do what we did today. So like you were there today, so you can do the paper, take your quiz, and go.
If we have like 11 o'clock lecture, do you think we'd be out there if we come to the 10?
I don't know. Yeah. So. Okay. Yes. Fit it in the best you can. I understand it's not perfect. Okay. So far, this is the list of stuff. I haven't added too much to the list you've been seeing. This is the same list that just grows after each lecture. The things I've added today were how do different enzymes know where to go, right? So that's the one thing I noticed while we were doing our review today. A lot of people were like, well, there's a hat. I'm like, yeah, but how does it get there? Why is it there? So you have to think to yourself, hats aren't just running around looking for nucleosomes to bind to. They're called to a place where they're needed, right? So there's that concept. DNA methylation, and then how readers and writers work together to establish stable repressive chromatin. And so people, one thing I noticed is that people were, this reminds me of a story, but I won't go there.
You have some.
Oh, you have some too.
I believe there was a lot of the story.
Oh, it's that protein thing. Right. Okay. So something that came up today is that people are thinking of readers and writers as separate from hats and other proteins we've been talking about. It's really just a way to think about hats and all the other chromodomain, bromodomain containing proteins that are involved. You can think of all your readers as things that recognize post-translational modifications on histones or modifications on DNA, right? They're reading that. And your writers are writing things. They're making marks on things. They're acetylating and methylating, stuff like that. So that's just a different one to think about it. If it's not working for you and it doesn't make sense to you, you don't need to go. I'm not going to ask you specifically about readers and writers on the exam. I just think it's a useful way to think about reading the histone code and reacting appropriately. So, Okay. So this is the last bit of stuff about the chromosome and nuclear architecture and its relationship to function. You've had this information for the last week. You read about it for the quiz 2 weekends ago. So it's, it's not completely new, hopefully. So there are three main elements required for stable inheritance of chromosomes to daughter cells. Stable meaning the chromosome DNA is stable, and that comes from telomeres. They keep the DNA end stable. Otherwise, you would have progressive shortening of your chromosomes. Inheritance, well, to inherit your chromosomes to the daughter cells or to pass them to the daughter cells, you need two things to happen. One is you need to replicate them, so you need an origin of replication. And the other thing you need is a centromere, because that's where your kinetochores are going to set up and your mitotic spindle is going to attach there and segregate your chromosomes that way. Okay, so those are that's right out of the quiz, right? Those are your three main elements that are required for stable inheritance. So I'm going to go through complementation assays a little. The reason I have this in my lecture is normally when we don't have snowstorms, this week we are doing a paper that is using complementation assays, and I like to introduce it. I'm introducing it anyways because I think it's a useful thing to think about, a way to think about how a lot of early research in cell cycle, research was done. Pretty much what you do is use a complementation assay to, what you have is some sort of mutant, a strain of yeast, it's usually in yeast, that has a mutant, which has a phenotype. That phenotype might be, remember, something you can see. It might be no chromosome segregation. It might be no, that's a really obvious one. It might be no cytokinesis, something that you can look at these yeast cells and say, this isn't happening. You've got that mutant, and then what you do is you put different bits of DNA in on a plasmid usually, and you're looking to see whether you can complement for the defect. You put something in, see if it fixes the defect. That's what a complementation assay is. So you're looking to, it's usually for gene identification. You're identifying a gene that's going to complement a functional defect in some mutant yeast strain. So when they first started doing this, they would have all these, they would do random mutagenesis. Now there's more targeted mutagenesis, but way back in the day, it was this random chemical-induced mutagenesis. Then they would screen the cells to find mutants that have different defects. They categorize these mutants into defects in different classes. These are cell cycle defects, for example. And then they would start to characterize each mutation and identify genes with different functions. Now it's way easier. Now there's so many sequenced genomes. You don't need to do anything like this, right? When you've got a sequenced genome, you just sequence your gene of interest, and then you blast it and compare it to other things out there. And you can start to say, oh, these domains are there. This is similar to this. And you can start to test things that way. But before we had these sequenced genomes and all these databases that we could just mine, this was the way things were done. And I think still people do use some elements of this. So some major concepts in complementation assays. First of all, you're using a mutant strain of yeast. And then there are different selection markers because you always need to keep track of the plasmids you're putting in. So for example, yeast, they can grow on really minimal media. They don't need much of anything because they can make most of their amino acids. And so for these assays, you use these strains that have mutations in biosynthetic pathways used for making histidine, uracil, and leucine. Those are your most common ones. And then they put the gene for those things that are mutated on the plasmid so you can kind of keep track of what's where. So your plasmid you might have a yeast strain that is His negative, so it can't make histidine, but on your experimental plasmid, you'll have the gene for His, and then you'll grow it on a minimal media that doesn't have any histidine in it, and that's how you'll be able to know that, okay, what you're looking for is actually there. I'll give you some examples shortly. And so you can do positive or negative selection using this. You need to have a phenotype you're looking for, otherwise it's not very useful because you have to be complementing something. So growth or morphology or something like that. And then this was historically used for identifying genes involved in the cell cycle. So there's three experiments, classical experiments that were done using complementation assays to identify essentially the origin of replication. It's telomere sequences and centromere DNA. And the paper what we would have done, I believe, was the centromere DNA one, I think. But, well, take a moment to cry about that. Okay. So for this one, in yeast, the origin's called the autonomous replicating sequence. It's an origin, right? Origin of replication, that's where DNA polymerase binds and starts replicating the DNA, right, during S phase. And so this is how they identified it through a complementation assay. So they had some kind of plasmid. And notice, I don't know how well you in the back can see it, but in blue it says LU. That's your selection marker. That's the gene for making leucine, okay? And then they're using the cell line that can't make leucine. So there's no way these cells will grow if they don't have this plasmid in it. That's just a selection marker. That's not part of the science. It's just a way to make sure that your cells have what you think you're putting in. Sometimes transformations don't work. You think you put your DNA in, it doesn't go in, you don't see a phenotype. You can't really do science if you don't know you actually got it in there. So that's why you use these leucine negative cells, and you put the plasmid containing the leucine, and then you grow on media that doesn't have leucine. And so there's two things here. They had two plasmids, a control, and then they had a plasmid which has this ARS, the origin of replication. Okay? And so they found that when you put plasmid, which contains nothing but the leucine gene in it, then you're not really going to get much growth. But if you put a plasmid that contains leucine plus the origin of replication, then you will have some growth. Not a ton of growth, because remember you have S phase and then M phase, so you replicate your DNA and then you segregate it to your daughter cells. Notice something's missing here, so you're not segregating to daughter cells, right? There's a centromere missing. But it is being passed to one daughter cell, right? So here you have your origin of replication. Here you don't. If you don't replicate your DNA, you can't pass it to anyone, so you can't get growth. Does that make sense? And so what they're trying to do is they're trying to identify what sequences are required for replication, and they're trying to see through looking at whether or not the yeast can grow. Right? Because every cell division results in cell growth, right? So this is how they identified that this origin of replication is required for plasmid replication. Okay? Any questions? I'm going to give you guys a minute. I just want you to take a minute, because this stuff can be really confusing and backwards to think about. But if you take a minute and just look at this and walk your way through it and talk to your neighbor just to try to get it, because there's going to be 2 more of these in a minute. I want you to do that, and then we're going to reconvene for the other ones, and I'll clarify any confusions, because I think complementation assays can be confusing. Okay, do you want to take a minute? Yeah. Think for a minute.
So,
Make sure that your plasmid is going to find out.
I think it's similar to gene therapy.
yeah.
If there's defects,
So they have different lines.
If an artist wants to do something,
So you basically just select
when you get that ERS,
Yes.
I think it's similar to what Snyder randomly knocking on the knees and what's wrong with it, why is it not doing it?
You're kind of cheating trouble. Yeah, Oh, right, okay, so not up here, over here. Okay, so what happened here is, so compare these 10 up here, here, it only went to that's like opposite.
Yeah, I can send you through whatever I got.
Now there was a 10 up here.
I mean, I got only 5.
Yeah, that's been on the yeah. Does it make it?
That's a screenshot.
I was thinking it was like the whole neighborhood was like right out. I like.
This air, not just here. In this case, because of the south, and in one case,
Yeah, Byron.
Okay, so I'm seeing questions are coming up. I'd like to give you guys a minute to think about this because now I'm starting to see lots of questions, which is better than me just flying through it and you guys going. So I'm getting lots of questions about the purpose of the leucine gene. That's a selection marker. For those of you who've done any sort of transformations into bacteria before of a plasmid, usually you have antibiotic resistance and then you grow it on a media containing the antibiotic to make sure that you're only growing bacteria that contain the plasmid. It's the same kind of thing, except for it's on an antibiotic. You're selecting with something else. You have these cells which can't make leucine, but they need it to grow. The only way they're going to be able to grow is if they get leucine from their plasmid. So it's a way to ensure that your plasmid is getting into your cell, because the experiment is comparing presence and absence of this origin of replication. That's what the experiment is. But if you don't even know if your plasmid got in, then you can't analyze your data at all. Okay, so that's one question. Hopefully that clarifies a little bit. Think about it a little more. You have a question?
Yeah, but this, so the one that says yes, with the one that it says yes and the other arrow says no, does that mean that there was no centromere?
Right. So the question is, how come here it's yes and here it's no? Right. There's no centromere. Here you only have the origin of replication. No centromere, no, well, telomeres are irrelevant in this situation anyways. But, right. So that's why, so you would guess, so I got another question here before, which is there two plasmids here? Yes, because here it replicated because of the ARS. But it couldn't segregate because there's no centromere, so it all ends up in one. So there's actually not much growth because only one out of every cell division grows. Okay? Okay, so I have a question up in the back.
Oh, I was just wondering, if it says, mitotic segregation is faulty, that it was just in reference to the plasmid, or if it was everything, but you answered that already.
So this really has to do with the plasmid. All that can really follow here is the plasmid because you can only grow with the leucine. Right. Okay, question, yes?
These mutations were made manually, like just knocking them off, or is it like you just didn't intentionally remove those like sequences?
For the leucine, you mean? Or you mean?
ARS, like ARS or telomeres or like all that.
So in this case, what you're doing is you're following the plasmid. So you've engineered, here what you've done is engineered it. So, because these are leucine negative cells, they're not ever going to grow unless you have a plasmid with leucine on it. So then you can follow its ability to grow based on the other things on it. So here, because you can't replicate this plasmid, it can't be passed down. It's like skipping S phase and trying to go to mitosis. Okay? Here, you can do S phase, but you can't actually do mitosis. Only 50% of each cell division gets it. Okay? So you can say, so the people who did this experiment, they were able to say this sequence that they've put in is required for replication of the plasmid. Okay? Any other questions? I'm going to go through the other two, and then I'll give you guys another minute to digest. Understanding the concept of selection can be sometimes a little tricky. So just hang on for a minute, and hopefully as we get through the next few, we'll start together. Okay, so here we're identifying yeast centromere DNA sequences. Again, using a complementation assay. And so here you've got plasmid, and what's different, you have your ARS, you need that to replicate your plasmid, and here they put in centromere DNA. And in this case, you have it going to both cells. You still have leucine negative cells, so you're sure that your plasmid's in there because they shouldn't grow at all if there's no plasmid in there. And they're going to 100% of progeny because you have a centromere. So if you can compare this, where it's only going to one, no centromere, to this, where it's not going anywhere, no origin of replication, so you can't replicate the DNA, to this, where it's going to both. So that suggests that when you add in these centromeric DNA sequences, that allows for chromosome segregation to both daughter cells. Okay? And so then, how would this look if you deleted your ARS from this, but you kept your centromeric DNA sequence? What would your result look like? Do you think you would get both, one, nothing? Nothing. Ooh, yay. Nothing, right? Because you can't replicate. You don't have your ARS. You can't your origin. You can't replicate. So that's like trying to do mitosis without going through S phase, right? Okay, good. Okay, so here they were doing, they were studying telomere sequences, and so you should say to me, well, plasmid circular, there are no telomeres, and so they basically did it. By actually cutting the plasmid with a restriction enzyme to make chromosome ends artificially. And what did they do? So here they added some telomere sequences to the end. So you've got a cut plasmid with no telomere sequences and a cut plasmid with telomere sequences. And here they saw there was no growth. You have your ARS, you've got your origin, you've got your centromere, but what's happening is what's happening to the plasmid?
Just shrinking.
It's shrinking, or it's basically not stable, right? So it can't, regardless of what it has, it's not stable, so it can't be segregated to daughter cells. And in this case, they added these telomere sequences, which stabilize it, and so it's got its origin, it's got its centromere, and it could be passed to both daughter cells. Okay, so do you guys want to take another minute to think about this, or do you have questions? Do you want to take a minute? Oh, yes. Yeah. Yeah. So, as with every experiment, you need your control and your experimental. Your control is just a linearized plasmid, and your experimental was adding in some telomere sequences. So, something to understand is that your DNA at the end of your chromosomes is not just called telomeres. It actually has specific sequences that make it telomeres. Make it telomere sequences. And so in this case, without the telomere sequences, you actually are getting no growth, right? And that's because that plasmid is unstable. So even though you have your origin and you have your centromere, it's just not stable, so it won't be passed to daughter cells. Where here, where they've added, you can't see it that well, but hopefully at home you'll see it. Right over here, they added in telomere sequences. And that stabilizes this linearized plasmid. And so it has everything else it needs, and so you have stable inheritance of your chromosome. Okay. Does anyone have any questions? Do people have any questions about just the overall concept of complementation assays, of the purpose of selection markers, of anything like that? Yeah.
So complementation assays to basically look at protein interaction.
Complementation assays, can you use them to look at protein interactions? Here we're not looking at protein interactions, right? Here we're actually looking at fixing a genetic defect by putting different genetic elements in. So it's essentially assigning gene function or DNA element functions, right? So there's a defect. So yeah, so some people during the quiz emailed me that somewhere they saw something about protein interaction complementation assay. Yeah, that's not what I was talking about. It was more about the genetic to complement a genetic defect. So it's like a genetic screen to assign a function to a gene or a fragment. In this case, it's fragments of DNA. Okay. Any other questions? Are we happy? Yes.
So I know you said mineralized, so would that mean if it doesn't?
Okay, so I'm just gonna expand on the point of linearizing just for everyone. So I just got a comment that was quickly taken back about linearizing the plasmid, and I just want to make sure everyone understands that you can't have telomeres if you have a circular genome, right? Telomeres are DNA ends. If you have a circular genome, you don't have telomeres. And so in this case, they needed to make it linear, just to kind of make telomeres. Okay, and by definition, it's not a chromosome. It's just a plasmid, so it's going to lack telomere sequences, and so they can really show it's unstable without telomere sequences, and then they artificially add it in. Okay. Okay. So that is how a lot of that stuff was discovered. So the centromere, as you already know, is a region of DNA that's required for chromosome segregation, right? You've got your sister chromatids, and your centromere is usually that part where it's condensed and attached. Okay. Your centromere DNA, its purpose, so it's actually DNA, and its purpose is to bind to proteins that form the kinetochore. And the kinetochore is the part that attaches to your microtubules, your mitotic spindle, and that's how you can segregate the chromosomes to the daughter cells. So let's go back to histone variants for a minute. There's actually a histone variant that I feel like I do a lot of picking up things. So SEMP-A is a histone variant that shows up and in basically in the centromeric region. So there's the centromeric DNA and it binds to SEMP-A. So if you did immunofluorescence and you did two stains, one was for SEMP-A and one was for histone H3, which is the normal histone you would see there. You would see histone H3 in green throughout the whole chromosome, but SEMP-A would be only present at the centromere. Yes?
I don't know if I'm going backwards, but when a histone variant enters the nucleosome and switches out of the histone, what actually tells it to do that?
So why are histone variants in certain places?
Yeah, and then not in other places of the histone.
So what do you think?
Maybe it's more condensed, like inner chromatin is more condensed and it has different.
So I'm not sure. So the comment up front was about maybe there's more heterochromatin there, and that's what brings it in. I think one is it's sequence specific in this case. So there's the centromeric DNA has AT-rich sequence, but it's histone chaperones that play the role of switching out histones for histone variants. Now, I believe in this case, SEMP-A is always there. It's not, because that DNA sequence is always there. It's not going to change. Your centromere is where your centromere is. I mean, there are neocentromeres that you may have read about in the textbook, but generally, there's a specific sequence of DNA. It's AT-rich, and it tends to bind to histones containing SEMP-A. That's all I got for you. You want more?
I mean, I was just more of like in other, like.
With other histone
with
variants?
other histones, like.
Yeah, so
and this is always there, but in other histones.
Right, so like H2AX, right? H2AX only comes with DNA damage, right? And so there you're going to actually sense DNA damage by sensing changes in DNA structure. There's going to be a recruitment of H2AX to those regions. There's going to be activation of histone chaperones. So there, depending on the variant, I guess you could say there probably is a whole process for bringing them to certain places. Yep. In terms of SEMP-A, it binds to the centromeric DNA, and that's sequence specific, except for neocentromeres, but that's a different story. And it's required for assembly of kinetochore proteins. So what do you think would happen if I feel like there's a lot of chit-chat. Does anyone have any questions? No? Sometimes I hear, like, random chatter, and it could be just like, oh man, this sucks. Or it could be like, I don't understand, and then I need to make sure people understand. We're all good? Okay, so what would happen if SEMP-A were overexpressed in the cell? What do you think might happen? What would be the outcome on the structure of the chromosomes or on mitosis, for example? Yes? Huh? You couldn't separate? Is that what you're saying? If you overexpress something? You think it might inhibit? So I hear maybe it inhibits. What did I hear?
I would say over including kinetochore.
Okay, so maybe there would be increased recruitment of some kinetochore proteins, maybe to the wrong places. What are you saying? I heard, saw your hand up. Nope. Down here? No. Anyone? Yep.
Can it affect genes that are like next to it and cause like position effect maybe?
So could it bind to a different place, perhaps, and maybe affect some genes that are close by? Is that what you're thinking? Maybe. So, yep.
So if there's more of those histone variants, they will try to replace the regular histones like out of it, then just like try to replace the whole thing with.
So do you think that maybe it would replace histone H3 in other places?
Yeah, I mean, it will like the whole DNA
Yeah, well, so that's a bunch of these kind of together, I think are correct. If you have lots of SEMP A, I would expect you would start to create other centromeres. And then what? If you have more than one centromere, what might happen? More chromosome segregation. But do you think, like, do you want multiple centromeres per chromosome system chromatids? You might, yeah, you might disrupt the structure. You might break your chromosomes, right? Yeah. Yeah, so that'd be bad. Yeah. So what do you think would happen if you have SEMP A deletion?
No sex
So
deletion.
you would only get one.
That's an obvious one. Okay, see, everyone's like, oh, no chromosome segregation. Right. No SEMP A deletion, no kinetochore would set up, no mitotic spindle would attach, no chromosome segregation. Right. What if there were mutations in your centromeric DNA and it went from AT rich to all G's?
Setting up their own place.
So it wouldn't bind SEMP A anymore. Right? And then but maybe SEMP A would then bind somewhere else and you would form a neocentromere. Okay. Cat.
So the function of SEMP A is to make this the centromere?
Yes, SEMP A binds to centromere DNA, and then the kinetochore binds to that, and then you segregate your chromosomes by binding your mitotic spindle to your kinetochore. Yeah. Okay, so some important points about centromeres. They require SEMP A. So you cannot have a centromere without SEMP A, meaning without SEMP. And as we saw in the complementation assays, without that centromeric DNA, you can't have chromosome segregation, right? Was everyone convinced? Some people are nodding yes. Some people are looking away. Don't look at me. SEMP A often binds to AT-rich alpha satellite DNA. Often, not always. There are neocentromeres that can form. And we won't worry too much about that. So another thing. I'm sorry. You gotta start throwing money. It's good. It keeps me limber. Okay. I was thinking about how much fun it would be if we did some sort of movement in this class, like exercise. I'm trying to walk more. Do a downward dog every now and then. Okay, so if you think about the type of DNA, I'm glad I'm talking about. If you think about the type of DNA present in centromeres, it's usually heterochromatin, and from that it follows that H3 and H4 histones, H3 and H4, tend to be hypoacetylated and hypermethylated, right? You would expect heterochromatin to be hypoacetylated and hypermethylated. That kind of makes sense. And then with that, you'd expect there to be low gene expression there. So that all kind of makes sense. Okay, so a quick practice problem. You're studying centromere formation and function. Using the information that you know now about centromere formation and function, interpret the following data. You're introducing different types of sequences into DNA and evaluating the following data. You're looking at segregation to daughter cells. You're looking at SEMP A binding and modification of local histones. And this is what you get. You get, so you look at you get three different things. You get no SEMP A binding. So remember, these are different types of DNA you're putting in, right? So you get no SEMP A binding, hypoacetylated and hypermethylated histones. You get no SEMP A binding, hyperacetylated, hypomethylated histones. And you get SEMP A binding, hypoacetylated, and hypermethylated histones. Which samples are likely to segregate to daughter cells? So 1, 2, and 3 are different DNA sequences that you put in. So take a minute to read this over and think about it, discuss and we will do other
Do they have to segregate
Well, you're talking about central government.
We should have I know you're anxious every time I in biology next year. You always get the day where you got questions. Boring material. All the essays are kind of the same. You want to talk about immunology? I'm right back. I think immunology is actually a lot of the I have a hard time. I just want to put it out. And he does a lot of walking. No, in your class.
Okay. Is that fun? Okay, lots of people, I like it when people, especially people I don't hear from in the back, are calling me over. Please call me over to tell me what you're thinking. Because you might not be comfortable raising your hand in front of the whole class, but usually you're comfortable just talking to me quietly. So always call me over, tell me what you're thinking. Okay, so who thought it was number 1? Raise your hand. Good. Who thought it was number 2? Good. Who thought it was number 3? Oh, okay, good. Yeah, it was fairly easy. The dead giveaway was the first part, right? So someone did say to me, oh, but it's hypoacetylated, hypermethylated, then you won't get any gene expression. Well, the thing to keep in mind is we're only talking about centromere formation. It's not the whole genome. It's only the little bit of centromere DNA. And I only share that with everyone because that's come up in years past where people get caught up on gene expression and centromere formation. I don't know why. So I just want to make sure that yeah, you have this heterochromatin in that region. And if you look at that, I mean, it looks even more condensed there. So I mean, it's condensed chromatin anyways because it's separating. So much for that. Okay, good. On to telomeres. Oh dear. Okay, on to telomeres. We're going to stop having fun and we're just going to plow through. Okay, so Nobel Prize in Physiology or Medicine, 2008. Carol Greider, I only point her out because I went to Hopkins and people, they beat you over the head with how fantastic Hopkins is constantly. You have to drink the Kool-Aid when you're there. We're just drink the Kool-Aid, drink the Kool-Aid. So we're all come out going, Carol Greider. So, but then there was also Elizabeth Blackburn, who was her PI at the time. She was a postdoc, and then this guy who no one ever told me about, which is real sad, because I was at Hopkins and we were all like, Carol Greider. But what really funny story, a friend of mine recently, she sent her daughter to Roland Park middle, in the Ingenuity program. And she emails me, she texts me, she's like, do you know Carol Greider? Her daughter is friends with my daughter, and they invited my daughter to go for a play date, and I don't know how I feel about her going over to this person's house because we don't know this family. And I'm like, she won the Nobel Prize. It's totally going to be fun. Don't worry. I mean, sometimes you do have to worry about your kids going places, but this part, don't worry about it. So I thought that was funny. I was like, oh my God. But the other thing that I thought was totally cool is that her daughter is in Baltimore City Public Schools, which was pretty freaking cool. My kids are in Baltimore City Public Schools, so I was like, woohoo, represent. Okay. Anywho. So that's it. Okay, so this is all off the Nobel Prize website. They do a really great job kind of explaining the relevance of what each Nobel Prize that's awarded. So feel free to go and check it out just about other things. Okay, so the early observation was that broken chromosomes were unstable, but chromosome ends appeared to be protected. And so why is that, right? Chromosome ends should look like broken chromosomes, right? They should start binding to each other and rearranging and fusing and all that, and that doesn't happen. So that was the question, is what is protecting the ends of the chromosomes? And so it was found that they're protected by telomeres, and they're kind of these caps at the end, and they contain a telomere sequence, which are these repeats. And they're species specific, and they're maintained by the enzyme telomerase. Pretty much. So I've got a video just to explain the end replication problem, because you got to ask yourself, well, why are they with each progressive division shrinking in the absence of telomeres, in the absence of telomerase? So we'll look at that video, and then this is a link. Elizabeth Blackburn gave a lecture. Actually, I think it's like a 3-part series of lectures on the function of telomeres and telomerase. And so people always are very interested in telomeres and telomerase because of aging, because of cancer. It's just kind of a really sexy topic. So I just put this link up here. I'll take you to YouTube, and you can, on a Saturday night when you're rocking out, you can learn about this. So let's see this one.
The ends of linear chromosomes pose unique problems during DNA replication.
Here, I'm going to make it big in a minute.
Because DNA polymerases can only elongate from a free 3' hydroxyl group, the replication machinery builds the lagging strand by a backstitching mechanism. The ends of linear chromosomes pose unique problems during DNA replication.
So this is just explaining the end replication
Because DNA polymerases can only elongate from a free 3' hydroxyl group, the replication machinery builds the lagging strand by a backstitching mechanism. RNA primers provide 3' hydroxyl groups at regular intervals along the lagging strand template.
The DNA sequence too.
Whereas the leading strand elongates continuously in the 5' to 3' direction all the way to the end of the template. The lagging strand stops short of the end. Even if a final RNA primer were built at the very end of the chromosome, the lagging strand still would not be complete.
And
the final primer would provide a 3' hydroxyl group to synthesize DNA, but the primers would later need to be removed.
Right,
the 3' hydroxyl groups on adjacent DNA fragments provide starting places for replacing the RNA with DNA.
And so you can imagine
However, at the end of the chromosome, there is no 3' hydroxyl group available to prime DNA synthesis. Because of this inability to replicate the ends, chromosomes would progressively shorten during each replication cycle. This end replication problem is solved by the enzyme telomerase. The ends of chromosomes contain a G-rich series of repeats, called a telomere. Telomerase recognizes the tip of an existing repeat sequence. Using an RNA template within the enzyme, telomerase elongates the parental strand in the 5' to 3' direction, and adds additional repeats as it moves down the parental strand. The lagging strand is then completed by DNA polymerase alpha, which carries a DNA primase as one of its subunits. In this way, the original information at the ends of linear chromosomes is completely copied in the new DNA.
And so that's why you keep having these same repeats, because those repeats, and we'll go into this in detail when we talk about telomerase later on, but those repeats are in your enzyme itself because it carries its own primer. Okay, so I always like movies, so feel free to watch that if you have any confusion about the end replication problem. But it's really about removal of that final RNA primer in your lagging strand. So this just explains it a little bit more, how you have to remove this primer, and then you've got this gap. And so there are a few different experiments that were done that led to these guys getting the Nobel Prize. So one is they were looking at, so basically they made these mini chromosomes in yeast, and they were normally damaged. But when they added in telomere DNA from Tetrahymena, another species, it protected the mini chromosomes. So that's how they were able to say, oh wow, these DNA ends, these telomeres, even from a different species, have this protective effect. Yes?
Yeah, but you said that it was telomeres are species specific.
Telomeres are species specific in the sense that every telomerase has its own sequence within it, its own primer that it carries, so they have its own sequence. But actually the protective effects worked across species. So so you can look at a telomere sequence and figure out which species it comes from. Yeah, I see where that could have very well. So then they did another experiment where they were trying to look for the activity, this telomerase activity. And so they went back into Tetrahymena and they took the cell extract, so they had all the proteins within the cell, and they added that plus synthetic telomere DNA plus radioactive nucleotides to see if they could extend the length of the DNA ends. And so they were able to show that you actually had increased length of DNA ends in the presence of this cell extract, suggesting that cell extract contains some kind of enzymatic activity that lengthened DNA. And so that was how they discovered telomerase. Okay? So, this is kind of what I said before. So this is where I'm talking about how it's sequence specific. Each organism has a different repeat sequence. And that's because telomerase itself carries its own RNA primer, which has its own sequence. And so telomerase comes and it extends this strand, and then it's filled in by DNA polymerase alpha on the bottom. Again, telomeres are lengthened on the leading strand by telomerase, and it's filled in on the lagging strand. So the question I have for you guys is, do telomeres need to be repaired in all cell types? I'm getting lots of no's. Why? Yes?
If you have cancer cells, they get to live forever.
Right, so exactly. In cancer cells, you have that happening, and they live forever. That's not good. Yes?
There are cells that need a high turnover rate, like skin cells, we would want them to live very long.
Cells that with a high turnover rate, you don't want them to live very long. When you have things that shouldn't live very long, that do live very long, then you get cancer. Yep.
Cells in G0 that don't replicate.
Cells that in G0 that don't replicate. Do you need to repair their telomeres? No, because they're not replicating, right? Right? You only need to worry about telomeres. You only need to worry about chromosomes shrinking in cells that are replicating, right? Does that make sense? Okay, so what kinds of cell types would have active telomerase? Huh? Well, yeah, cancer cells for sure. That's one of the defining characteristics of cancer cells is often dysregulated, overactive telomerase. Yeah. Germline cells. Yep. Huh? Yep, germline cells, exactly. Cancer cells. Okay. So in human somatic cells, telomere repeats act to prevent unlimited cell replication. So it's like a molecular measuring stick. Cells are born with a certain number of telomere repeats, and after a certain number of cell divisions, cells will enter senescence. So they'll stop dividing, and that's a good thing. Do you think telomerase is on or off in these cells? Who says on? Who says off? In these cells. So these are these cells that enter senescence. Who's saying they're off? Who's saying?
If one part is on and the last part is off when you're dying.
Okay, so you have a certain number of repeats, right? And with each round of replication, it's shrinking. And then once you kind of run out, you stop dividing. So would you say telomerase is on or off in these cells? Off, right, because otherwise you would keep extending those repeats. So it's kind of a way to control the amount of cell divisions each cell has, which is one of the reasons that primary cells don't last very long in cell culture. But if you can immortalize them by activating telomerase. Yeah.
This is kind of what makes up
The what? Oh, I don't know. I feel like I'm going to misspeak on that. What was the question? This is the Hayflick number.
This what contributes.
Yeah. I feel like I can't speak on that. Do you want to expand to the rest of the class?
Oh, the Hayflick limit is just basically the number of positions the cell can go through before it completely stops dividing.
Right.
And I was wondering.
Okay, so then in that case, absolutely. I think I just don't think of it with that name. Yeah. Yeah. Okay, yeah. So there are these time cells that we used to use in the lab, and they're telomerase immortalized microvascular cells. And so there are these microvascular cells that were immortalized by adding in and basically keeping overactive telomerase on. Okay. So there's actually a feedback mechanism that allows yeast cells to control their telomere length. And so there was an experiment that they talk about in the textbook where they put cells, they put chromosomes in with really long telomeres and with really short telomeres, and then over multiple divisions, they all end up with the same length telomeres. And so there's a way of regulating telomere length in these cells. What protein do you think might be being regulated by this feedback? Telomerase, right. Okay, so I think this might be the last slide. Telomere binding proteins regulate telomere length. So they actually bind to the telomeres, and they also prevent activation of the DNA damage response. And the DNA damage response can often be activated by broken DNA and double-strand breaks. And without these telomere binding proteins, the chromosome ends might look like damaged DNA or a double-strand break. And if you activate the DNA damage response, that's not good when it's not needed, because you can get cell cycle arrest and apoptosis. Any questions about this stuff? Okay, so in the last few minutes, I guess we've got about 15 minutes, I just want to show you really quickly what we did in recitation today. And this is going to be posted on Blackboard. Oh, people are leaving. So what I had everyone do, and what I'd like you guys, those of you who have not been in recitation, what I'd like you guys to do is draw the following. Not right now, you don't have to in 15 minutes. I'm going to try to walk you guys through it a little bit now so you can see what I want. I want you to draw open transcriptionally active chromatin with those following things, with more than 3 nucleosomes, and showing all post-translational or DNA modifications. Don't worry about copying this because it's going to be on Blackboard, that are likely to be present, types of proteins likely to be associated with this chromatin, types of domains that those proteins might have, and then the regions of this chromatin that might be DNA nuclease sensitive. And the same for repressed or silenced chromatin. And so, let's see. Okay, let's see if this is going to work. So what I'm looking for is you guys to actually draw out, I always like to draw my, I'll move my hand away in a minute. I always like to draw my nucleosomes like that. So you draw multiple nucleosomes and put in your histones, right? And so I want to know all those things that I just said. So post-translational or DNA modifications. So you're going to have your active chromatin. What kinds of post-translational modifications would you expect to see? Acetylation. So where would acetylation come from?
Transcription factor.
Okay, so a transcription factor might bind and recruit what kind of protein that's going to acetylate? Some hat, right? And then that hat is going to maybe acetylate this this lysine on the histone tail, right? Or it might acetylate some lysine on the histone core. And so what's going to happen to this acetylated lysine? What's it going to do? It's going to recruit a bromodomain containing protein, right? I'll put a B there. And that bromodomain containing protein might recruit what kind of protein? Okay, so a lot of things. It might recruit a nucleosome remodeling National Republican Convention. A nucleosome remodeling complex. And then there's this acetylation at the histone core. What's the purpose of that? What's that doing? It might be creating a binding site for something. What else might it be doing? Changes the charge on the lysine. Remember the positive charge of the lysine, so it can't interact with the DNA, and so it starts to loosen it up. So loosen DNA. So you've got that, and so then what else? So you might have another histone tail that's also acetylated by this hat. What might that be recruiting? It might recruit another hat, which might have a bromodomain in it, right? And so what might that hat do? Maybe it'll acetylate this histone, and then so on and so on. So what if this was previously silenced? What other protein might, so maybe this histone tail will recruit something else that might help with demethylating. So you might recruit a demethylase, which is going to remove the methyl groups. Okay, so that's kind of the thing I want to see. So this is active. If you've got repressed, you're going to see something similar but completely the opposite, right? So you're going to have, again, oh wait, so what part is DNA sensitive in the active? Right, so all that is DNA sensitive, right? Anything not bound by proteins. So if you've got this repressed chromatin, what kind of modifications are you going to see on it? You'll see methylation. Okay, so where's that methylation coming from? From where? Where's the methylation? What enzyme is methylating it? A methyltransferase, a histone methyltransferase, or a DNA methyltransferase? Maybe. In this case, histone. So maybe there's some kind of transcription factor or a repressor binding site here that's going to recruit a methylase, a histone, not methylase, methyltransferase. Right, and it's going to methylate, or it's going to methylate, right? And what's that methylation going to do? So it'll recruit a protein containing chromodomain, maybe an HDAC. That's going to remove what? Right, so it's going to remove acetyl groups, right? Because you don't want acetyl groups if you're repressing. And if you, okay, good. So what else might, so you've got that happening, you'll have more methylation happening. So what else might be recruited by a methylated histone tail? A what? DNA what? A DNA methyltransferase. That's going to methylate the DNA. Where is that DNA being methylated? On the cytosines. What happens if you mutate those cytosines? They won't be methylated anymore. Right. Okay, so I'm giving you the gist of what I want to see. And so doing something like this can be really useful for taking all the stuff that I've given you and putting it into something that is big picture-y. So I want you guys, you Thursday people, to try to work on this a little bit before recitation because I don't know how much time we're going to have. And hopefully I can go over it with you guys and the questions.